Hi Year 12s. Today we're going to be looking at the last video or the last PowerPoint in our DNA and proteins topic. So in this video we're going to be looking at genetic engineering. The PowerPoint will be split into two different videos. So the first one is this one on genetic engineering and the second one will be on CRISPR. So last two videos for cells and proteins with the test coming up in about a week or so. Let's get started. So first off, genetic engineering. So basically, once we discovered we had the ability to sequence DNA, and we knew the role that the base sequence of DNA in genetic information, truthfully it was only going to be a matter of time before humans decided that we could create the ability to combine DNA from different sources and try and play God as such. There's many different names for genetic engineering, such as recombinant DNA technology, genetic modification, genetic engineering. All these names mean the same thing. Basically, it's just combining DNA in test tubes, inserting DNA, that new recombined DNA, into cells, where the new DNA will be expressed via protein synthesis. What's the actual process of genetic engineering? Well, first off, cell membranes are broken down. The nuclei is separated through a process of centrifuging, and centrifuging is just spinning very, very fast. We don't have any. We do have a centrifuge machine at school, so we might see this process in class. We then treat the nuclei with special chemicals which break down the nuclear membrane. Once we've done that, we can isolate the DNA from the nuclear proteins. Restriction enzymes are then used to pinpoint and cut DNA at exactly specific points to help manipulate the DNA further. We use something called sticky ends, and we'll talk about sticky ends in the next slide or two. But know that the DNA, after they've been cut with restriction enzymes, are creating sticky ends, or short lengths of DNA with sticky ends. So, actually let's talk about sticky ends now. So, there's two different types of ends. You can have blunt ends or you can have sticky ends. Blunt ends would just be basically cutting a DNA straight through the side. So if you were cutting right here, a blunt end would be this DNA segment ends with CG. A sticky end is when you've started the cut in one strand, then you've gone up and done the cut at a different point in the other strand. That leaves this section of exposed nucleotides on each side, and that's sticky because they're willing to bond with other fragments of DNA because they have this end that's looking to make hydrogen bonds. Okay, so sticky ends, basically just when you cut a strand of DNA at different points on each strand, therefore creating strands with extensions that are looking to bond with other strands. There's lots of different restriction enzymes that can do this. The one in this diagram here is EcoR1, which is an enzyme that we take away from E. coli, hence the ECO. Okay, and R1, restriction enzyme 1. And it will always look for the code GAATTC. So that's what we're looking for here, GAATC. And it will always cut between the G and the A. So here it cuts GA. Then it moves along again to find the next GA and cuts between those. Okay, always leaving TTAA as the sticky end. Either switched around either way. Okay. A sequence is recognized by a restriction enzyme. That's called the restriction site. So when we're looking for this GAATTC, that is the restriction site, and it cuts at the G and the A. Okay. Generally, the restriction site's about 4 to 6 nucleotides. Now, like I said, some restriction enzymes will cut straight across the strands, leaving a blunt end, but others will cut horizontally, like we've just described, creating sticky ends. Restriction enzymes generally have evolved in bacteria, and bacteria use them in a way to fight off viruses. We've isolated a large number of restriction enzymes from different strains of bacteria, so that's where we're getting our restriction enzymes from. We're basically isolating them from bacteria, and bacteria are just using them to fight off viruses. So how do you identify and isolate the right gene? So once we've extracted the DNA from cells, as we've talked about in the last slide or two, and we've isolated it away from nuclear proteins, restriction enzymes are coming in, they're cutting them into smaller fragments. But you then have to find the exact right fragment that contains your specific gene. And there's two different ways that we're going to talk about how that's done. So the first is using a DNA or RNA probe, and the second way is using the antibody method. So we're going to look at both of those in detail now. So first, DNA and RNA probes. So in order to use a probe, we basically need to know the short sequence of nucleotides in the gene in advance. So we can only use a DNA and RNA probe if we know the sequence of the fragment that we're looking for, or the sequence of the gene that we're looking for. If the protein, so we don't necessarily have to know the exact sequence, but if we know the protein that the gene's coding for, we can work backwards from the amino acid sequence and actually deduce the nucleotide sequence of that gene. So you either have to know the exact nucleotide sequence or know the protein that the gene codes for. If you knew either of those, you can use DNA and RNA probes. What is a DNA and RNA probe? It's just a short segment of single-stranded DNA or RNA that has the corresponding sequence of bases to bond with the gene that you're looking for. It's radioactively labelled and then will be mixed with the double-stranded DNA fragments that contain the gene you're looking for. The solution will be heated, breaks the hydrogen bonds between the DNA strands, and then it will be cooled to allow bonding to reoccur. While it's cooled, some of the probe molecules will actually bind to those DNA fragments, and we can use the radioactive label to identify the probe and therefore locate the gene that it's attached to. So that's how we select the right gene for genetic engineering. Another way is the antibody method. So this is one that we would use if we don't know the nucleotide sequence, but we know that we know what the protein is the gene codes for. We've taken a protein, the gene that we're looking for codes for, we've inserted it into an animal, that animal creates antibodies, we then collect those antibodies and label them radioactively or chemically, and then we're cutting up the actual DNA that we're wanting to test with into different fragments. We take those fragments and we put them into bacteria using recombinant DNA technology. Each bacteria will only take about 1 or 2 fragments, and those bacteria will then be incubated into colonies that will have that DNA, and they'll use that DNA to make the protein that we're looking for. All the colonies will be treated with labelled antibodies. The antibodies will only work on the protein that their antibodies are focused on. So therefore, all we need to do is look for the colony that's actually being treated with the antibodies, and then we can figure out, alright, that bacteria had this gene inserted into it, or this fragment with this gene inserted into it, therefore this must be the fragment that codes for this protein. So it's working backwards basically. So once we've isolated and identified the gene that we're looking for, we can actually clone that gene with the use of bacteria. Bacteria contain plasmids, which are small rings of DNA separate to their bacterial chromosomes, so separate to the nucleoid. We actually have the ability as humans to remove those plasmids from bacterial cells, and we can cut them up with restriction enzymes, making sure we leave them with sticky ends. We can then choose a fragment or segment of human DNA using the probe or antibody method and place that cut gene into the DNA plasmid. Okay, and they'll bond together because of their sticky ends. DNA ligase would be used to incorporate the gene into plasmids and rejoin them into rings. The plasmids will then be mixed with the bacterial cells, and those bacterial cells will take in the plasmids if they're grown under the right conditions. Once they take them in, every time the bacterial cells reproduce, so with ideal conditions every 20 minutes, that gene will be copied. That's how we clone genes. We use this process in insulin production. So we take the human gene for insulin production, we cut it with restriction enzymes, we take a plasmid from a bacterial cell, we cut it with the same restriction enzyme, we then use DNA ligase to connect and rebuild that DNA plasmid into a ring, and we put the ring, or the DNA plasmid, into a culture of bacteria. The bacteria take in that new DNA plasmid containing the gene for human insulin. We make the the bacteria reproduce, and therefore we copy the gene for human insulin. So that's one example of how humans clone. Transgenesis. Okay, so whenever we take or create an organism with DNA from another organism, we call them a transgenic organism. Okay, and the process that we use to put the new gene into another organism is called transgenesis. So basically gene from another organism. Lots of different ways that we can do transgenesis. So we can use microinjections, we can use vectors, and we can also use TI plasmids. TI plasmids. So TI plasmids are generally going to be used in plants. So we're going to basically be introducing a gene into a plant. Basically, a TI plasmid is something that's found in agrobacterium. An agrobacterium is a bacteria that causes tumours in plants. So we remove the TI plasmid from that bacterium and we add the desired gene to it. We then return that plasmid to the bacterium where we use it to infect the plant. The bacterium will insert the TI plasmid into the plant cells and cause those cells to divide rapidly, producing a tumour. We then physically remove that tumour, cut it into many pieces, treat it with tissue culturing techniques to produce new plants who contain the desired gene. Viral vectors. Viruses can introduce genetic material into a host cell. That's how viruses function. Generally, that will cause that cell to manufacture the viral proteins rather than the cell's own proteins. We can actually take a virus and modify that virus to, instead of containing its own genetic information, it contains a desired gene. So it's no longer got the ability to harm the host, but when it enters the host, it tells the host cell, create this gene, create this protein, rather than the protein of the virus. That, in turn, incorporates that gene into the DNA of the cells that we're injecting the virus into. Microinjections. So this is one that's used only in animals, where we insert the gene that we want to insert into another organism into a fertilized ovum with a microinjection. The ovum is held in place, and a glass micropipette passes through the plasma membrane of the fertilized ovum and into the nuclear membrane. The desired gene is then injected via micropipette into the nucleus, and the resulting animal therefore contains that gene. The only problem with microinjection is there is not a 100% success rate.